Bismillah ar-Rahman ar-Rahim. In this lecture, I'm going to explain the glandular epithelium. The epithelial cells that specialize for produce and secrete the various macromolecules called gland. The gland can be divided in two groups by the number of the cells. When we have one cell, we call it the unicellular gland. More than one called multicellular gland. The well-known example for unicellular gland is our goblet cells. In this picture is a section from the small intestine villus. You can see the goblet cells. We call it the goblet cells because the shape of the cells is very resembled to the wine goblet. You can see in this picture. These cells produce the mucus in the surface. The mucus is a viscous material, it's a slimy liquid that is composed mainly by mucins. Mucins is a glycosylated protein, it's a glycoprotein. When absorbed water, converted to the mucus. The mucus usually doesn't stain by routine staining, I mean H&E, because of the carbohydrate in the glycoproteins. You can see white part in the apical part of the cell. We call it the unicellular cells because you can see just one goblet cells among the other epithelial cells that aren't secreting cells, that just they are covering epithelial cells. Also, you can see a schematic picture from the goblet cells. You can see accumulation of the mucins or mucus in the apical part of the cells, and the nucleus of cells also located in the basal part of cell. All of the multicellular glands originated or developed from the surface epithelium. You can see here the epithelial cell proliferate and grows into the underlying, usually connective tissue or underlying tissue. Sometimes these epithelial cells remain and keep their connection with the surface and make exocrine glands. By this way, the end of the bottom of the cell, bottom of this accumulation converted to the secretory cells, and the connection also converted to the duct. So the material can transfer by this duct to the surface in the exocrine gland. But sometimes after proliferation and growing into the underlying connective tissue, the epithelial cells lose their connection with the surface. Sometimes also these cells immigrate a long distance. For example, thyroid gland originated from the end of the tongue, but they immigrate from this place into the front of our neck. After immigration, these cells comes near to the blood vessels, and their secretions absorbed by these blood vessels and transfers to the target cells throughout the body. We call this kind of gland endocrine gland. This gland doesn't have any connection with the surface, so they have to release the material into the blood vessel. Also, all of the glands packed and protect by connective tissue. I mean the connective tissue comes around the glands and enclose all part of the glands and make a capsule for this gland, and they usually protect them and make a separated organ in our body. I said each exocrine gland have a two part. One part is a secretory portion and another part is a duct. You can see the secretory portion somehow in brown to red in this gland, and then we have a duct in yellow, and the small duct fused together, attached together, and finally make a main duct or excretory duct in this exocrine gland. We can also classify the gland according to the shape of secretory portion and the shape of or structure of the duct. Here we want to classify the gland according to the structure of the secretory portion. You can see we have two kind of secretory portions. Sometimes we have a tubule or tube-like structures. We call this gland tubular gland. And sometimes we have a round or sac-like structure. We call it acinar. Acinar means like a grape. You can see it's very resemble to the grape. This part is a secretory portion and this part is a duct. So the gland, the exocrine gland can be a tubular or acinar. Also here we have a tubular and acinar. And also in this slide you see we have a tubular gland in this part, branch tubular, cold tubular, and also acinar and branched acinar. Again, here we have a branched tubular, branched acinar, and also sometimes we have both of them together, tubulo-acinar. Another the classification for the gland, as I explained, was according to the structure of the duct. You can see in this gland. You can see in this gland we have just one duct. When we have an unbranched duct, we call it a simple gland. And when we have branched duct, we call it a compound gland. Because of that, we call simple tubular, simple branch tubular, simple cold tubular, and simple acinar, and so on. And here you see compound tubular, compound acinar, and compound tubulo-acinar. In this slide, I have some example for each type of gland. For example, the simple tubular, the gland in the large intestine, or a simple acinar, the gland in a urethra. And also for a compound, for example, Brunner gland in the duodenum, or a compound tubular, or pancreas is a compound acinar, and salivary glands and lacrimal gland are also the example for a compound tubulo-acinar. Another classification of the glands is according to the mechanism of secretions. Some of gland or epithelial cell in gland accumulate the secretions in the apical part of the cell, and when the epithelial cell is stimulated, these granules fuse to the cell membrane in apical part and just release the material. We call this kind of also secretion exocytosis, but here we call them merocrine. Also, we have other epithelial cells. These cells accumulate the secretion in granules, and when the cell is stimulated, these granules comes to the apical part and detach from the apical part of the cells with a small amount of cytoplasm, also with a cell membrane. We call this kind of gland apocrine gland. And the last groups, the cells accumulate materials inside of the cells until the cell die by this accumulation, and the whole of the cells detach from the glands and release into the out of the gland. We call it holocrine. So by the mechanism of the secretion, we can divide the glands in three groups: merocrine, apocrine, and holocrine. Let's see in also these three kind of glands in the gland. You can see here a gland with a holocrine secretion. You can see the whole of the cells detach from the epithelium and comes out of the glands. In the merocrine, you can see just only the materials secreted out of the gland. And in the apocrine, you can see these materials with a small amount of apical cytoplasm and cell membrane detached and come out of the glands. Let's see some example for this gland. The merocrine, commonly most of the glands secreted by the merocrine mechanisms. For example, here you can see the salivary gland. Three of them, the submandibular, sublingual, and also this salivary gland. All of them are merocrine gland. Also the pancreas, also the liver, all of them are merocrine gland. For the holocrine gland, just we have only one gland by these mechanisms, and that is the sebaceous gland in the skin. The sebaceous gland, you can see the cells, the whole of the cells detached and released into the out of the glands, and in our skin produce the fat for our skins and make our skin fatty. And the example for an apocrine gland is one of our gland is an apocrine, and is mammillary gland. The mammillary gland, by this way, by the apocrine mechanism, they release a lipid inside of the milk. By the lipid part of the milk produced by this way, and the proteins and water and other materials produced by a merocrine secretion. The epithelial cell of the mammillary gland have two mechanisms to produce materials. One apocrine for release lipid, and merocrine for release proteins. And also here you see a real picture from the sebaceous gland. You can see in these parts the cell have a nucleus and start to accumulate the lipid inside of the cells. When the cells reach the apical part, you can see the cells, the whole of the cell detach, and the cell died. We don't see any nucleus here, and after that the cell detach and release into the duct. And also here we have a part of mammillary gland. The epithelial cell release a lipid by a small amount of cytoplasm by apocrine mechanisms. And as you know, the epithelial cells are renewed continuously in our body because they are in the surface in the surface of our body and should be replaced very fast. And this renewal, the rate of this renewal is different in our body. Sometimes, some place we have very fast renewal, some place we have a very slow. For example, in the small intestine we have a very fast replacement, and in the large gland we have a very slow replacement. Here you see one gland from the small intestine and also a hair follicle in our skins. This renewal is because of the stem cells between the epithelial cells. Sometimes these stem cells scattered between the epithelial cell. For example, in this intestinal gland, you can see the stem cells in red. These stem cells immediately proliferate and differentiate and replace every epithelial cell in the small intestine. And in some places in our bodies, stem cells are located and gathered and aggregate in one place called a niche. The niche of stem cells. For example, here in a hair follicle, each hair follicle has a bulge, and this bulge is a place for a aggregation of a stem cell. Call it the stem cell niche. From these places, stem cells migrate to the outer part of the epithelium, then proliferate and differentiate to the epithelial cell of the skin, even to the hair follicle cells and also the hair cells. And at the last slide we have three clinical words: metaplasia, carcinoma, and adenocarcinoma. Metaplasia is a transformation of one epithelium to the another epitheliums, or changing of one epithelium and the epithelium from one type to the another types. For example, in the smokers, the respiratory epithelium, pseudostratified columnar epithelium, gradually change into the stratified squamous epitheliums. Or another example for metaplasia, it's in the individuals for chronic vitamin A deficiency. In these persons, the epithelium of urinary bladder, again the urothelium, change gradually into the stratified squamous epithelium. Carcinoma is a type of cancer. The epithelial cells get cancerous, especially the covering epithelium, not the glandular epithelium. And adenocarcinoma is also cancerous tissue originated from the glandular epithelium. So adenocarcinoma from the glandular epithelium and carcinoma from the covering epithelium. For example, the skin cells get cancerous. And example for the adenocarcinoma, the pancreas cells get cancerous. Thank you for your attention.